So you've heard a lot about T regulatory cells in the molecular perspective. Now you're going to hear about regulation in a broader sense. So just to give an overview of the talk, I'm going to give an introduction to FDA Oncology, an overview of FDA Expedited Development Pathways. I'll talk a bit about recent trends in next-generation oncology trials, the changing companion diagnostic paradigm, and I'll close with a discussion on novel endpoints and approaches. So just a brief word on the FDA and our mission. We're responsible for assurance of the safety, efficacy, and security of drug products. I'm housed in the Center for Drug Evaluation and Research, drugs, monoclonal antibodies. There's also medical devices in the Center for Diagnostic and Radiologic Health, biologics like CAR T cells, which you heard about yesterday, vaccines, blood products, and CBER, the Center for Biologic Evaluation and Research, and many other responsibilities: food, cosmetics, veterinary products, tobacco. It's estimated that our regulatory body accounts for about 25 cents for the dollar spent by Americans. We don't take cost or payment into account. I think you'll hear more about that later from Dr. Lowe. And we don't regulate the practice of medicine. Just a brief word on our Office of Hematology and Oncology Products. We've engendered a lot of goodwill over the years. We come to these meetings and people like Dr. Chabner says, wow, the FDA has really changed its ways. And I think a lot of that is due to the able leadership of our skipper, Dr. Rich Pazder. This is one of his initiatives. We restructured our office a couple years ago to make it more align it to academic models. So we have disease-specific groups within our various divisions. I'm the clinical team leader for thoracic and head and neck cancer. And we have all different disease teams. And I think this has led to more consistent advice, more of a expertise, individualized expertise on the various diseases within the agency. We've also initiated a scientific liaison program, which has helped increase outreach to key stakeholders, including professional organizations, other government agencies, patient groups, and academia. So this is the regulator's dilemma, the delicate tightrope dance that we have to endure on a daily basis with multiple slings and arrows fired our way. On the one hand, be flexible, efficient, and interactive. On the other hand, consistent, thorough, and independent. On the one hand, we have to be cautious, but we can't be too cautious. We can't stifle innovation, and we can't have too many regulatory hurdles. On the other hand, we can't be too cavalier because that could lead to toxic deaths, delayed safety findings, and the notion that FDA is asleep at the wheel. So moving on to our expedited development pathways. Broadly speaking, you can think of approval pathways in two general terms. We have the traditional or regular approval pathway. That's based on an improvement in a direct measure of clinical benefit, how a patient functions, feels, or survives. So it's a longer life, a better life, or an established surrogate thereof. So what's an established surrogate? You could think of decreasing blood pressure to decrease cardiovascular morbidity or mortality. And then we have the accelerated approval pathway. This was initiated in the early 90s due to the HIV crisis, where viral load was used as a surrogate endpoint. And these are based on improvements in surrogate endpoints reasonably likely to predict clinical benefit over available therapies. You need post-marketing studies to confirm clinical benefit. Failure to confirm clinical benefit could mean that your agent is withdrawn from the market, as was seen with bevacizumab in metastatic breast cancer several years ago. We use this tool, accelerated approval, frequently in oncology, using surrogate endpoints like response rate and progression-free survival. So this is just our four major expedited programs. I'm not going to talk about fast track or priority review. I've already talked about accelerated approval. I'll walk you through breakthrough therapy designation, our newest kid on the block. So what is breakthrough therapy designation? It was signed into law in 2012. Actually, Dr. Chabner had a nice piece in The Oncologist talking about the backstory about how this was made into bipartisan legislation. one of the few pieces of bipartisan legislation in that Congress. And it's for serious and life-threatening illnesses like cancer. It's for drugs where, based on preliminary clinical evidence, shows a substantial improvement over available therapies. We've taken great advantage of the breakthrough therapy program. About 40% of the breakthrough requests across the Center for Drugs have been in oncology, and about a third of these have been granted. In 2014, of the 9 new molecular entities approved in oncology, 5 were breakthrough designated. And this pace has only accelerated in 2015. I don't have the exact numbers, but I suspect perhaps it'll be about what we saw in 2014, or maybe even a larger sample size. So with breakthrough therapy, with any new program, there are opportunities and there are challenges. The opportunities is that they bring an all-hands-on-deck mentality, including senior leadership, for these transformative, knock-your-socks-off, know-it-when-you-see-it therapies. It aligns and prioritizes key FDA review teams across clinical, statistics, manufacturing, clin-pharm, toxicology, and inspections. And it optimizes communication between the FDA and the sponsor. However, there are some challenges. When granting breakthrough, we get a lot of requests. Every company thinks they have the next breakthrough. What is the right threshold for granting a breakthrough designation? Do you want to be more lax, or do you want to be more strenuous? What constitutes available therapy? We've taken the position that a drug that has accelerated approval is not available therapy. So if you have your next ALK inhibitor in line and it shows really durable response rates, and you have an accelerated approval for a drug in the ALK crizotinib refractory setting, that could potentially be eligible for breakthrough. How late is too late in the game? We've approved breakthrough therapies after the phase 3 trial has reported out. Really, the spirit of the legislation was for early clinical results, but we found it useful at times to sort of mobilize forces, particularly from the manufacturing component, and sort of align key agency components. Resource saturation is an issue. It's an unfunded mandate, and it just creates more work. On what basis should we rescind a breakthrough? We haven't had to do this yet in oncology, but it's a possibility. And then finally, manufacturing timelines can be a bottleneck, because even if your drug shows knock-your-socks-off activity in phase 1, sometimes the manufacturing is not quite up to speed with the clinical data. So here are some examples of breakthrough therapies granted and subsequently approved in my field, lung cancer. Ceritinib for crizotinib refractory ALK-positive non-small cell lung cancer, based on an expansion cohort in phase 1. The first in human to accelerated approval took a little bit over 3 years. It was a very compressed review timeline as well. I think Dr. Chabner wrote a piece calling it "Ceritinib runs the 3-minute mile." And as highlighted in our review documents, which is publicly available on drugs.fda.gov, so you can look this up, we did have a late manufacturing issue for an early intermediate that popped up late in the review cycle. And I think breakthrough really helped in mobilizing. We went to our center director, and it helped mobilize our forces. We were able to dispatch an inspector to that site in a foreign land to expeditiously resolve this manufacturing issue. Other examples would be the PD-1 inhibitors. Recently, just a couple weeks ago, we approved pembrolizumab in PD-L1 second-line non-small cell lung cancer on a validation set in a phase 1. I'll show you the schema of that study in subsequent slides. And nivolumab for non-squamous lung cancer, which was a very compressed review timeline. So next, I'll talk about recent trends in next-generation oncology trials. So this is the traditional drug development paradigm, as well as companion diagnostic paradigm. It's very linear, it's sequential, it takes a long time, over a decade, a lot of money, billions of dollars, to develop a drug from validation all the way to FDA approval. And this is the vision, right, in 2020. It looks a lot like those Krebs cycle metabolic pathways you saw earlier. More of an iterative, adaptive credentialing of drugs and companion diagnostics. These are some of the challenges with what I term as the old paradigm. If you look back in lung cancer and many other cancer types in the last couple decades, we've had a lot of phase 3 failures. And basically, you take a heterogeneous, unenriched, unstratified patient population, you give 1,000 patients, randomize them to platinum doublet versus platinum doublet plus drug X, and the risk you run is a high risk for phase 3 failure or clinically small effect sizes. In this new era, it's nice to talk about this in Mass General because this was one of the pioneering centers where you take an enriched patient population of ALK rearranged lung cancer, for example, give a targeted therapy. You might see large clinically meaningful effects in a small patient size. The problem becomes as you get to lower and lower slices of the pie, say 1% prevalence of even a common cancer like lung cancer, the number needed to screen becomes prohibitive. Furthermore, as I'll explain in subsequent slides, the one drug, one biomarker per trial is unsustainable. We need to move towards common multi-analyte platforms, and we need more rapid learning, failure, and confirmation. So this slide is just to show that the low-hanging fruit has all been plucked. The common, more frequent driver oncogenes, BRAF in melanoma, EGFR in lung cancer, are all accounted for. And now we have this long tail of potentially actionable mutations. If you add up this long tail, it's actually more common than some of these frequent mutations. But as of now, these genetic aberrations are variants of unknown significance. So recognizing all these problems, I think the field has moved and converged to these master protocols. These can be multi-drug, multi-biomarker trials, sometimes involving multiple pharmaceutical and diagnostic companies. They can be broadly stratified into umbrella trials or basket trials. Umbrella trials test the impact of different drugs on different mutations in single types of cancer. Examples would be the BATTLE-1 and 2 studies in lung cancer, the I-SPY-2 in breast cancer, and the LungMap in lung cancer, which I've been very involved in, and I'll describe further in subsequent slides. You can also think of a basket trial where you test the effect of a drug or drugs on a single mutation or a bin of mutations in a variety of different cancer types. The first one, to my knowledge of this kind, was an imatinib basket trial, which was a multinational study, which actually led to FDA approval of multiple small subsets of PDGF-driven tumors. There's also BRAF, like vemurafenib and dabrafenib baskets. One was recently published in the New England Journal. And of course, the NCI MATCH study. So the LungMap is a unique public-private partnership, a collaboration between FDA, the NCI, the Foundation for the NIH, and the Friends of Cancer Research, as well as the National Clinical Trials Network. And this was the original LungMap design. Basically, patients with second-line squamous non-small cell lung cancer would get screened with a broad-based NGS platform, the Foundation Medicine platform. And based on their molecular profile, if they were PIK3CA mutant, they'd be assigned to a targeted agent versus standard control, CDK4/6, FGFR, and HGF. If they didn't have any of these matches, they would be randomized to a PD-L1 antibody versus docetaxel. Well, fortunately, things change rapidly. At least recently, they have been in lung cancer. And when the study was launched in the summer of 2014, subsequently, we had new information. First, in November, Amgen announced termination of development of relotumumab, which was an HGF MET inhibitor in gastric cancer, which effectively closed down this HGF arm. And in March 2015, we approved nivolumab in second-line squamous non-small cell lung cancer based on the results of the 017 study, which showed an overall survival benefit over docetaxel. Incidentally, we approved this 3 months ahead of the ASCO presentation, as well as 3 months ahead of the New England Journal publication, which I'm not sure how the investigators felt about it, but we felt it was important to get this out to the US public. In light of these changes, this is the new LungMap trial schema, which hopefully all of these amendments will be implemented soon. On the MATCH sub-studies, instead of doing the randomization to docetaxel, which is no longer a palatable option for patients, it's actually single-arm studies And based on what they see in the single-arm phase 2 component, if they see a knock-your-socks-off type durable response, maybe that would be registration grade in and of itself. If you see more of an intermediate type signal, if feasible, maybe there would be a randomization to a standard of care as yet to be defined. So taking into account that standards of care can change. And then in the non-matched sub-studies, there's a checkpoint naive, combining a CTLA-4 and a PD-1 versus PD-1. And in checkpoint refractory setting, there's also PD-1 CTLA-4 combination arm. And NCI and the cooperative groups are not the only people getting into this master protocol game. Pharmaceutical industry is also starting their own trials, usually not sharing drugs with each other, but within their own portfolios. This is an example of the KEYNOTE-1 with pembrolizumab. Started off as a phase 1 dose escalation study, and then multiple amendments, multiple new hypotheses looking at different tumor types, different biomarker test set, validation set, different doses and schedules of pembrolizumab. And ultimately became a 1,200-person trial and very successful in that it led to 2 accelerated approvals, one in melanoma, one in lung cancer. So what's next for master protocols? Probably, my guess, my crystal ball would be, these next generation of protocols will probably have more comprehensive omics profiling, potentially whole exome, proteomics. More novel combinations will be necessary. We're starting to see this with the lung map, with the PD-1 plus CTLA-4. And from an agency perspective, we're starting to think about, should we provide some guidance on best practices for these expansion cohorts and master protocols, both for investigators, IRBs, DSMBs, as well as best practices for statistical methodologies. So next I'll discuss the evolving companion diagnostic paradigm. As I stated earlier, as an agency, we're starting to move away from this single test, single drug paradigm to credentialing multiplex platforms and potentially using multiple drugs on the same platform. This makes a lot of sense for many reasons. One, for example, in lung cancer, now tissue is scarce and EGFR and ALK are actionable. Potentially we'll have ROS and BRAF soon as actionable mutations. And given tissue scarcity, cost considerations of repeat biopsy, it just makes more sense to have a multiplex assay. Also for drug developers and diagnostic developers, it doesn't make sense to reinvent the wheel every time to find a new companion diagnostic partner to test one analyte every time you want to develop a new drug. Finally, of course, for researchers to test hundreds of aberrations at the same time makes a lot of sense for scientific inquiry and discovery on resistance and responsiveness. So recognizing this, and as part of the President's 2016 budget to launch the Precision Medicine Initiative, the President earmarked funding for both the FDA and NIH to develop these large, well-curated data repositories. We think this makes a lot of sense to try to standardize platforms so that your readout from Illumina is the same as Thermo Fisher, is the same as Foundation. Get a better handle on genotype, phenotype, and on actionability. We'll need to figure out how to incentivize data sharing because this will be critically important. We can't just operate in our silos of academic centers anymore. Another concept I wanted to present, this is sort of hot off the presses, would be the companion diagnostic versus complementary diagnostic paradigm. Companion diagnostics, well established. If you go to our website, we now have 22 drug companion diagnostic pairs. And the definition is that it's a test that's essential for the safe and effective use of a drug. Complementary diagnostic doesn't really exist as of yet from a regulatory perspective. I think it was just an offhand remark by Liz Mansfield at a meeting, and it sort of snowballed. But potentially the definition could be, is this a useful test to identify patients more or less likely to derive benefit from a drug? So this is a case study, a recent case study I'm intimately familiar with. Pembro versus Nivo in non-small cell lung cancer. Pembro was approved with a companion diagnostic. It received accelerated approval for only that PD-L1 high, 50% or greater, based on durable response rates. It was only studied in PD-L1 positive patients, and the companion diagnostic was required and is part of the indication on the drug label. They use this DACO 22C3 clone, IHC, and it's a 50% score. In contrast, nivolumab did not have a specific PD-L1 eligibility criteria. They did have a pre-specified analyses looking at different 1, 5, and 10% PD-L1 expression cut points. And it did suggest that actually the overall survival data was potentially driven by the high expressors. However, we didn't want to limit the indication because there were a population of PD-L1 negative patients who were deriving benefit. It was a head-to-head trial against docetaxel. It was less toxic than docetaxel. And there were responders in that negative cohort who had durable responses. They used the DACO PD-L1 28/8 antibody. And in their press releases, when we approved the drug, they said this is the first complementary diagnostic approved. So stay tuned. This leads to a problem where you have a highly competitive market. You have four PD-1, PD-L1 inhibitors very far along in development, many still being developed. You have different diagnostic companies, different cell types being measured. Genentech's looking at tumor and immune cells. The other companies are just looking at tumor cells. Different cutoffs, different clones. We recognized this, and we organized a workshop with AACR and ASCO on harmonizing companion diagnostics. And out of this formed a working group with the ISLAC, AACR, and industry to try to cross-validate these companion diagnostics. So finally, I'll move on to novel endpoints and novel approaches. These are our 3 traditional oncology endpoints. I'll start with the gold standard, overall survival. You need bigger and longer studies. It's a direct measure of clinical benefit. It's very objective. It can be confounded by crossover and subsequent therapies, and it can't be assessed in single-arm trials. Moving on to progression-free survival, it's less of a direct measure of clinical benefit, and it can be more subjective. You can be subject to ascertainment bias-informed censoring. It does account for stable disease. We've always taken the position that magnitude of PFS is very important because of some of these limitations, and it can't be assessed in single-arm trials. And finally, response rate, which is making somewhat of a resurgence, was heavily used in the 70s. We went away from it with highly toxic chemotherapy, but now it's making a comeback in the targeted therapy era. You have smaller, quicker studies. It's a binary endpoint, yes or no. Doesn't account for stable disease. Can be assessed in single-arm studies because tumors don't naturally regress absent the drug in general. There are some barriers to randomized controlled trials. There's feasibility in low-frequency populations, crossover impacting overall survival assessment, and ethical issues when the intervention is highly active or the comparison is toxic. This came to the fore during the Dr. Flaherty's and others' vemurafenib studies where people questioned whether it was ethical to randomize against DTIC and BRAF mutant melanoma. So when are randomized trials unnecessary? Some have postulated the parachute test should apply when you have a large effect versus a historical control, like penicillin for pneumonia, insulin for diabetes, multi-agent chemotherapy for testicular cancer. But we do have to remember our history, and we always have to remember some cautionary tales out there, like autologous stem cell transplant for metastatic breast cancer. So we convened a working group to look at this with the Friends of Cancer and Brookings. I'd refer you to this publication. NCI, Dr. Simon, some patient groups, some pharma representatives. And we postulated that non-randomized trials could be used when the mechanism of action is supported by strong scientific rationale, preclinical data, drugs intended for a well-defined patient population. The drug produces substantial durable tumor responses, and benefits outweigh the risks. This is a meta-analysis we did in lung cancer, looking at the last 10 years of submissions, 14 trials, over 14,000 patients. We saw that response rate is strongly associated with progression-free survival on a trial level in lung cancer, which suggested to us that if you have a very big effect on response rate, you'll also probably have a very big effect on PFS when compared to chemotherapy. Finally, I want to touch upon novel metrics for response. So we see all the time at ASCO, AACR, these waterfall plots, these swimmers plots, these spider plots to try to quantitate, better capture response metrics. Some of the challenges with immunotherapy are that response rates in unselected patients can be modest. PFS curves separate late. There's this probably pretty rare phenomenon of pseudoprogression. What we've seen in lung cancer is the patients who do respond to immunotherapy have very durable response. In the case of Nivo, 17-month median duration of response versus 6 with chemotherapy. These are some of the progression-free survival curves. And what you can see is median PFS. Most of the patients actually don't benefit from these drugs and come off therapy rather quickly. There is a tail to the PFS curve, so about 20% of patients have a long progression-free interval. Those are the patients most likely, in this ADNO study are PD-L1 positive, deriving the benefit and driving the survival curves. These are the survival curves. Again, this is the 057 study, very mixed heterogeneous population, so it actually appears that docetaxel is doing better early, but then nivolumab clearly looks better. And then in the squamous population, actually the curves separate early and appear to continue to separate after the medians. We'll have to see if this pans out with further follow-up. So this Dr. Flaherty alluded to as well. This is where we need to be. We need to push up the plateau to have patients with more durable remissions. And we need to potentially look at novel endpoints to better capture how we're going to assess this in clinical trials. So there are things we can do. I think we're going to have working groups to look at some of these analyses, maybe landmark analyses for PFS or response rate or overall survival. And we have both ongoing efforts, both internally and collaboratively, to analyze completed trial sets for improved metrics beyond traditional RECIST. So it's been a busy time in lung cancer. These are just some of our approvals, a lot published in the Oncologist, and we have many more advances to come. I just wanted to acknowledge Dr. Pazder, as well as my thoracic and head and neck team, and I want to put in a shameless plug for our lung cancer Twitter chat. Thank you.
I promise I'm going to ask you a question. So
Go ahead, Adrian.
So can you tell us more about this new CDRH initiative? Because first of all, it could be used as a salvage of a negative study. So basically, you have a primary endpoint, you fail, but then you can go back and see whether something will help you. Do you think that the agency will accept something like that?
You mean the complementary diagnosis?
Complementary, yeah. And what the other issue is that it will take the incentive from industry to go to the traditional way of approving, devices as opposed to this one. So I'm still digesting, and I don't know if it's a good thing. And is it helping patients? I mean, in a sense, with this complementary for the Nivo. I mean, it's
Well, yeah. So I'll address your second question. I mean, is it helping patients? I think we had a lot of struggles whether we should restrict the indication. Clearly, if you look at the forest plots in that 057, clearly the PD-L1 positive patients were sort of driving the survival. As I explained, there were reasons why we didn't want to limit it to, there could be patients who are negative who were benefiting. But it could be useful still to test for PD-L1 positivity in second-line lung cancer, even for clinical trial purposes. Clearly, the negatives don't respond as well. Can we add new CTLA-4, OX40, LAG3, you know, different ways to enhance the immune response. So I think it can be helpful for patients, this complementary diagnostic. But yeah, I agree with you. I don't know what the downstream consequences. I think, you know, the companion diagnostic guidance will still stand that if you're going to enrich your population, you need to submit a contemporaneous application for your diagnostic to get your drug approved. So I don't think
although response is not a surrogate for survival for these drugs, appears to be not to be.
Well, the responders, if you do responder analysis, the ones who respond, you know, those are the ones who appear to be, you know, driving the survival.
And do you know the explanation why, you know, with these drugs, PFS is not significantly different as opposed to survival? I mean, this is something I can't understand. I mean.
Yeah, so I think, as I showed you, the median PFS, I think most of the patients don't benefit. The ones who do benefit a lot. So it's like 20% of the patients, at least in lung cancer, driving the survival. So 80% of the patients don't benefit. So that's why your median survival is no different from docetaxel.
So you think it's a measurement issue, not necessarily that. Okay, that's also okay. Thank you.
Since you mentioned the consensus panel of PD-L1 staining, when is a consensus statement expected? Is there? It's
a good question. I mean, so, you know, for these preclinical collaborations between industry partners in a highly competitive environment, it's very challenging to sort of herd the cats. I think there will be, so they wanted to only look at analytic components. They didn't want to tie anything to clinical outcomes. I think there will be a report, hopefully within the end of the year, for this blueprint, this year, yeah.
Gideon, thank you very much for a tremendous overview. And the only thing I missed from it was, and I know that the understated nature that you always hold, is the great commitment that the FDA has. I'm speaking now principally in the oncology division, to really communicating early on and collaborating, of course, in quotation marks, because we have to consider the sponsor is obviously separate. And the thing that strikes me is that this, especially in the breakthrough therapy legislation, that was only as good as the FDA chose really to make it. And you've made it superbly well. So I hope that in the future, one of the things that Bruce and I are talking about, we'll be working on, is really with the Society for Translational Oncology, convening, perhaps with friends as well, Friends for Cancer Research, a meeting with industry to actually discuss the pharmacoeconomics. As these trials are racing through, blurring the heretofore catenated phase 1, 2, 3 trials, going as you have in rapid review and hopeful when appropriate approvals, that great savings in time translates into savings, tremendous amounts of savings in money. We like to actually put some decimals on that and identify approximately what they are therefore saving and how those savings appropriate to society as well as to their shareholders can then be more allocated. So it's really the first step the government is making in helping stem this escalation in really very expensive drugs for patients who really need them. So thank you. Salute.
Thank you, Marty.
As the drug development time is decreasing, companies are really benefiting from that in terms of longer exclusivity for the drugs. That should be translated, I think, into some moderation of the increase in cost, which is now, you know, concerning so many of us. So
one would hope.
At what point will the FDA get into the business of talking about cost effectiveness?
Well, you know, I think when we make our risk-benefit determinations, I actually think it's a very good thing where, you know, cost is a separate discussion. So, you know, I have sort of mixed feelings about getting into that business.
You can't make policy statements.
And I can't make policy statements, but just as a reviewer standpoint.
It's on everyone's mind.
Absolutely.
Yeah. Okay, thank you very much.